Hi, my name is Taibu Oluwatosin, popularly known as Deep Blue. I'm here to take a video presentation on central dogma of molecular biology, which involves three different aspects. So the first aspect I'll be talking on now is replication. So this is the central dogma of molecular biology. It involves three different processes. First one is replication, followed by transcription and translation. So we are going to know what all this is all about as we go on. But for the purpose of this presentation and this video, I'll be focusing more on replication. Now, before we go into replication, I would like to review the structure of DNA. The DNA is double-stranded, as you can see, and it has different base pairs. Basically, our adenine, thymine, guanine, and cytosine. Now, it is very essential for us to know the base pairing. Adenine binds with thymine. As you can see here, adenine binds with thymine with the help of 2 hydrogen bonds, while guanine binds with cytosine with the help of 3 hydrogen bonds. This is very, very essential for us. I also want to remind ourselves that our DNA runs in 2 different directions, 5' to 3', and the other runs from 3' to 5'. Please, let's keep this at the back of our mind so that as we go through the process of replication, it will be easy and understandable. Now, the process of replication is the synthesis of new DNA from a parent DNA, and the process is semi-conservative. What does that mean? It is semi-conservative in the sense that in the new daughter cells produced from the DNA, it has an exact strand from the parent and an entirely newly synthesized strand. Now, the process of replication occurs in the nucleus of cells, and this particular process occurs before a cell divides. The DNA of a particular parent has to be copied before a cell divides. So in layman's word, we can liken the process of replication to photocopy. So DNA is copied during the S or synthesis phase of interphase during cell division. Of course, we all know the process of cell division, mitosis, and the likes. The process begins at origins of replication. What are these origins of replication? It is a sequence that are rich in the adenine and thymine nitrogenous bases. So these 2 strands open, forming what we call replication fork. As we go on, we are going to understand what the replication fork is all about. Now, active DNA synthesis occurs at the replication fork. In other words, we can say the replication fork is the point where DNA synthesis occurs. Now, DNA unwinds at short distances, creating the fork as shown in the diagram below. This is the parent DNA. Then this is the replication fork. You can see that the strands of the DNA have unwinded, forming replication fork. So we said this particular point is where active DNA synthesis occurs. Now, synthesis of DNA is bidirectional. That is, one goes from the 5' to 3' direction, and the other goes from the 3' to 5' direction. So this gives rise to 2 strands, which we call leading strands and lagging strands. What are leading strands and lagging strands? Now, the strand that is being synthesized towards the replication fork is known as the leading strand, while the lagging strand is a strand synthesized away from the replication fork. So as we go on, we are going to see different images that explain that better. Now, this is another image of the replication fork. You can see the leading strand we just talked about. It is going towards the replication fork, while the lagging strand is moving away from the replication fork. So several lagging strands give rise to fragments which are known Okazaki fragments. You can see that the particular DNA has been unwinded at a particular distance, forming the replication fork. Now, there are several enzymes involved in the process of replication. The very first enzyme is helicase, from the word helix. It binds to the DNA double helix and unwinds the strands by melting the hydrogen bonds between the nitrogenous bases. When we talk about nitrogenous bases, we talk about adenine and thymine, guanine and cytosine. So helicase melts the hydrogen bond between them, therefore separating the DNA strand. Now, after this DNA strand has been unwinded, the single strand binding proteins will attach themselves to each of the separated strands, and to keep them from rejoining, or we can say it keeps them from reassociating, or it keeps them from reannealing. So that's the function of a single-stranded binding protein. It's an important enzyme in the process of replication. Now, sometimes during the process of replication, when helicase unwinds the double helix structure, there is a creation of what we call supercoiling. Now, this supercoiling is not a good situation because it causes topological stress. So the topoisomerase enzyme relieves the DNA of supercoiling, thus preventing topological stress. It does this by creating a nick. When we say a nick, synonyms we use are cut or a gap. Yeah, there are two types of topoisomerase. We have topoisomerase 1 and we have topoisomerase 2. So as we go on, we are going to see the mechanism of action of that topoisomerase. Now, prior to the synthesis of new DNA, there must be RNA primers present to start the addition of new nucleotides. Primase is the enzyme that synthesizes and lays down the RNA primer. What we're trying to say here is that the synthesis of new DNA requires an RNA primer. The synthesis of the primer actually makes the action of our DNA polymerase easy. Okay. Now, DNA polymerase synthesizes new DNA by adding nucleoside triphosphate. When we talk about nucleoside triphosphate, we mean our ATP, adenine triphosphate, our GTP, our guanosine triphosphate, and the likes. Now, in prokaryotes, there are three types of DNA polymerases. We have DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. While in eukaryotes, we have 5 DNA polymerases, which include DNA polymerase alpha, DNA polymerase beta, DNA polymerase gamma, DNA polymerase delta, DNA polymerase epsilon. Of course, they all have their functions, their distinct functions they play in the process of replication in eukaryotic organisms. Now, DNA polymerase 1 is a repair enzyme, and it has a 5' to 3' and 3' to 5' activity. While DNA polymerase 2 is also a repair enzyme, but it has 3' to 5' exonuclease activity. When we talk about exonuclease activity, we are talking about the removal of nucleotides from the end of the DNA. Then the DNA polymerase 3 is the main synthesizing enzyme in prokaryotes. Of course, the DNA polymerase 1 and polymerase 2, which we say they are repair enzyme, they actually come into play when there is a mismatch or when there is a damage to a particular DNA. Now, like I said earlier, we're going to talk about the action of the topoisomerase. You can see here that this is a supercoiling of the strand. Now, this is the topoisomerase enzyme. It comes to relieve this DNA from this supercoiling. What does it do? It creates a cut on the strand. This is what happens. So after creating a cut, it therefore helps the DNA from having topological stress. So the supercoiling presence in the DNA molecule has now been relieved. That is the action of topoisomerase enzyme, and it is a very essential enzyme in the process of replication. Now let's look at all the enzymes in general. This is the overview of DNA replication. First of all, we talked about our helicase enzyme. This helicase enzyme, you can see, it cuts and separates the DNA. It cuts and separates the DNA strands by melting the hydrogen bonds between the nitrogenous bases. Now, whenever there is a supercoiling, the action of the topoisomerase come into play. It relieves the DNA from supercoiling. Now, when the strands have been separated, you see here that a single-stranded binding protein come into play, and they prevent the separated strands from reannealing or reassociating. Now, we'll talk about the RNA primer. This is the RNA primer. It is very, very important as it kickstarts the process of replication. So the DNA primase is the enzyme that actually lays down the primer. Now, let's talk about our leading strand, our lagging strand. You can see the direction of the leading strand. The leading strand is towards the replication fork, while the lagging strands is away from the replication fork, which gives rise to our Okazaki fragments. So we'll talk about our DNA polymerase. DNA polymerase is the synthesizing enzyme of replication. So these processes come into play whenever a cell wants to divide. So the DNA of a particular cell has to be copied or has to be replicated. So that is why replication takes place. So the next tutorial we're going to talk about the process of conversion of the DNA into an RNA, which is the second phase of our central dogma. Let's quickly go back. You can see the second phase of our central dogma is transcription. So it is very, very important for us to know all the enzymes in replication: the helicase, the SSBP, the topoisomerase, the primase enzyme, the DNA polymerase, and the likes. So thank you for listening.